Hello, and a very warm welcome to this Select Science Roundtable event titled Ask the Experts, Addressing Challenges in a New Era of Flow Cytometry. My name's Charlie Carter, and I'll be moderating today's roundtable discussion, and I'm delighted to be joined by a panel of flow cytometry experts, including Bill Telford, Derek Davies, Jodie Kroger, Mirko Koselli, and Bob Borderas, who will explore how to overcome common flow cytometry challenges in the lab. Please submit your questions for the Q&A session at any time during the webinar using the button to the left of your screen. And so, without further delay, I will hand over to Bob to introduce our presenters.
Great. Thanks very much, Bob, and thanks for BD and Select Science for inviting me to speak here today. It's a great pleasure. I've only actually got one slide here today. If we click to that one, it's a little bit primitive art, I suppose, but it's going to just describe a little bit like Bob was talking about, the sort of journey that we have to make in the cytometry lab. And if you think about it, this hasn't really changed in the 30 plus years that I've been doing flow cytometry. Somebody, this sort of gender neutral stick person on the left hand side, will have a question, and hopefully flow cytometry can give them the answer. And that question could be quite simple, and it certainly was simple 30 years ago. We might just be looking at the expression of one marker or the expression of a fluorescent protein. Or it could be a very complex question. We know now that flow cytometers are capable of looking at 20 or 30 or more different analytes. But at the end of the day, you're asking a question and you're getting an answer. But again, if we click, we'll see that this is an iterative process. Generally, it doesn't work first time. And the rule of thumb is if it works first time, it'll probably never work again. But in terms of what we provide from a core facility, we can have several inputs into this journey. And the first of those, if we click, is training. We want to train our users in a core facility. We want you to be self-sufficient. Flow cytometry is a very transferable skill. It also means that you can use the lab 24/7, and it means you're not knocking on my door asking how you switch this machine on. But it's not all about training. Sometimes, if we click again, we have the machine operation. Not everybody has the time, possibly even the ability, to run a flow cytometer, particularly cell sorting, particularly high-level experiments. So one of the key roles of a core facility is to actually do that, is to run the sorter and so on. But both of those are really aimed at getting some data files out at the end of the cytometer. If we click again, this is where the collaboration starts. The user and the members of the core facility will work together very often on the analysis of the data. What metrics do we need to get out of those data files to answer the biological question? And sometimes that might be just training somebody how to use an analytical program. Sometimes it's much more collaborative, and we've seen that has become more the case in recent years. Now, as Bob said, I started off using a BD fax analyzer. And if I look at that today, if I could find one, I would still recognize the components of today's flow cytometer. There's still a fluidic system, there's still some light sources, there's still some optics, and there's still some form of data readout. And those basic components have not really changed, although the individual parts, as Bill will come on to in a moment, have changed. So all of the inputs that we have into people's experiments have also changed in the last few years. So if we click, we'll see in terms of the training, it's not just about flow cytometry anymore. We've seen the introduction of imaging flow cytometry in the last 10 years, mass cytometry and imaging mass cytometry in the last 5 or 6 years. The last couple of years, the introduction of spectral analyzers. And every time a new bit of hardware arrives in the facility or in the market, we as core facility staff will have to understand how that works so that we can transfer that knowledge to our end users. Similarly with reagents, every time a new fluorochrome is developed, we need to understand its excitation, its emission, how well it plays with other fluorochromes, how stable it is, how it reacts to fixation, and so on. So it's a continuous learning process. Similarly, we click again there with imaging, with the machine operation. New technology requires new skills from the staff, much of which is done internally, but we do rely very much as well on the machine manufacturers. Now, I think if I looked at those changes in the sort of hardware that have happened over the last 30, 35 years, there's not been a sort of step like that. It's been relatively gradual. But I think one area that has changed, and here we're addressing challenges in the new era, is in terms of data analysis. So if we click again, we'll see that the way we do our data analysis is changing. It's not just as simple as looking for our CD4 expression or a cell cycle expression. If we're doing 20 plus markers, we have to think about the analysis in different ways. So we've seen the rise of high parameter analysis, clustering methods or dimensionality reduction methods, and even the introduction of machine learning and artificial intelligence over the past couple of years. And maybe that's been a little bit of a steeper curve than the hardware. You notice on the right-hand side, there's a little bit of a gap. So if we click, that will be repopulated or populated. And this is where our other cores come in. Again, I think this is something that's been more important in the past few years. We need to understand where our cells are going when they're coming out of the flow facility, particularly after cell sorting. And many of them are going to sequencing or other microscopy facilities, LM or light microscopy or electron microscopy. We have to understand more about those allied technologies. We need to know how to best prepare samples for single cell sequencing, for example. And the sequencing guys also need to understand something about how we've prepared the cells for them. So nothing is ever taken in isolation. Well, it probably never was, but even more so now, I think we have to be much more collaborative, because at the end of it, we click, there's a prize, isn't there, at the end of it? And if we're lucky, it's a big prize. Not all experiments are going to lead to that, of course, but I think many people would settle for just a renewal of a grant or a publication or even a good lab meeting in the end. And the very last thing I wanted to say, it's a very quick run through here, but the very last thing I wanted to say in the final click is that in the core facility, we're also thinking about how we can assess the training and the quality of the output that we give to our end users. Maybe that's slightly out of scope for today. So I'll stop there. And if anybody's got any specific questions, we'll come to that in the Q&A. And we'll also talk in a little bit more detail about some of those changes. So thanks very much.
Sure, absolutely. Okay, well, thank you, Derek. That was a terrific talk. So if I can have the first slide, please. Now, as Derek said, putting together a flow cytometry study experiment is a process. I'm going to talk a little more practically about all the steps, the tools that we have available for this, both for panel design, but also for trying to minimize compensation, which is probably one of the biggest challenges we face in flow cytometry. So this process begins well before you do your experiment, back when you're actually designing your instrument, picking out your what machine you're going to be running on. And as Derek said, the technology, although it's improved incrementally, it has dramatically changed in the last few years. The filters and dichroics we now use in our instruments are better. Using multiple lasers to reduce spectral overlap is a well-accepted approach now to attempt to reduce the amount of compensation between fluorochromes, as well as do more colors. We have instruments with increased laser power, which may or may not affect compensation. The detector types are changing. I think BD now has high sensitivity PMTs on their instruments, factory set PMTs, where you don't go in and adjust a voltage, but they're set at the factory at optimal linearity and sensitivity are now becoming more common. And we now have solid state detectors that are starting to be introduced into instruments, avalanche photodiodes, SIPMs that may be more linear. We also have to set up our instrument properly. Once you've got the instrument in the laboratory, doing quality control, as we all know, is very important. And doing some sort of technique to determine the optimal linearity and sensitivity for your detectors, such as a voltration, is also key to reducing the compensation that you see in your experiment. Now, there are a lot of anecdotal strategies out there like detector balancing. Some of these may work, some of these may not. We can talk about more about them in the discussion. Next slide, please. We also have now tools that will help us design good panels. And a lot of these are not really automated. They're simply following good panel design guidelines. If any of you did the BD Biosciences Horizon World Tours, this was covered in details, where we try to match our markers to brighter dim fluorochromes, phenotypic versus modulated markers, to try to maximize the information we'll get out of our panel and have it be each of the colors be as compatible as possible. There are staining index tables out there for both fluorochromes and CD markers. So not just for the fluorescent probes we use, and there are many of them now, but also the 300 plus receptors that we're going after on cells as well. Most of the major manufacturers are providing these. Spectral viewers are something I'm sure you're all very familiar with. Most of the manufacturers, reagent and instrument, have these online. They do give you percentages of overlap, but remember, these are not strictly compensation, although they can be related to it. And these spectral viewers are not always so useful for looking at multi-laser systems. We are starting to see better automated panel design tools, however. FluoroFinder is one that many of you are probably familiar with. It is a platform agnostic panel, so you can use it with any instrument platform. Prateep at NYU has designed Color Wheel, which is attempting to take all of these factors, fluorochrome brightness, marker intense markers, expression strength, into account in designing good panels. So we're getting good tools for that. Next slide, please. And finally, when the rubber hits the road and you actually have to do your experiment, most of us currently use our spectral overlap matrices, our compensation matrices, to determine whether our fluorochromes are working well together. However, as most of us have learned, you can have high compensation values and not necessarily have bad data. Compensation is only one of many factors that you have to take into account when designing a good panel. Many of us are now using so-called sample spread matrices, SSMs, which determine or attempt to determine the amount of sample spread, data spread, that is occurring in our population. This can in some ways be a superior metric for determining fluorochrome compatibility than standard spectral overlap, although they are both important. However, they are not perfect predictors. You can have a high sample spread and a relatively low sample spread value and vice versa. So there are new tools being developed all the time. I think at the next Select Science seminar, Mirko will be talking about some new algorithms for improving the use of sample spread to determine whether two fluorochromes are compatible or not. So basically, there are a lot of tools available, both way back at instrument choice and design, all the way up to running your experiment itself. But this is a complex issue, and there are no single silver bullets for it. You have to take all of the tools we have at our disposal into account to design good panels and to try to reduce the spectral overlap as well. I know we'll be talking in the discussion a bit about spectral cytometry, which is a big topic now. And although it's certainly in some ways more forgiving than traditional flow, the same panel design rules apply, and the same principles we're describing here apply as well. There's nothing magical about it. We still have to take those factors into account. So I will stop there, and we can move on. Thank you very much, and thank you to Science Select and BD Biosciences for setting up these seminars.
Thanks, Bill. Really appreciate it very much. If we can bring up the next slide. Sorry to everyone, we lost a little bit of the video feed from before. It's my pleasure now to introduce Jody Kruger. And Jody graduated from Xavier University in Cincinnati, and as many of us had an opportunity to start her flow cytometry, future in a laboratory at Procter & Gamble, where she was first introduced to the technology. From that time, though, many years ago, Jody joined one of the most prestigious, cancer institutes in the world today, and she worked, all the way through the entire process. And today, she's the director of that program. So it's my pleasure to introduce Jody for the next and the last talk. Thank you.
Thank you, Bob. Thank you for the invitation for me to participate, and thank you to Science Select for hosting this event. Also, thank you to my fellow panelists. I'm honored to be included in such a wonderful pedigree of experts, and I'm humbled by the experience, so thank you. To all of you attending, good morning, good evening, good afternoon, wherever you hail from, and thank you for joining us. As Bob said, my name is Jody Kruger. I've been a core facility manager now for 24 years at the cancer center that I work at. And my first introduction to flow, as Bob mentioned, was at Procter & Gamble, where I got to see a FACS scan, a 3-color FACS scan. I was responsible for purchasing my first caliber, 4-color caliber in 1995 there. And from there, I was fortunate to take on a wonderful role as a manager of a cancer center flow cytometry facility here in Tampa, Florida. My most recent acquisition is a 5-laser 64 detector or a spectral cytometer, and I've had all of the BD and Miltini instruments in between. So I've had a lot of experience with technology and the advancements, as you can imagine. So today's title, Addressing the Challenges in the New Era of Flow Cytometry, is near and dear to my heart because we certainly have a lot of them. The theme, however, that we're really trying to promote here is the basic principles and the fundamentals of flow. Because no matter how much we advance in these technologies, we have to have a firm grasp on our fundamentals in order for those experiments to work, as Bob alluded to earlier. Derek did a wonderful job of showing us again the user side of the journey through this process. We've all been through it, whether we're the person with the question or the facilitator of the person through that journey, through the learning process of flow cytometry, whether it's a 3-color traditional flow cytometer or a 64 detector spectral analyzer. The more things change, the more that they stay the same. So Bill introduced some fairly high-level concepts that are still fundamental to flow, regardless, again, of the complexities of the experiments that you'll run. They can be a little terrifying to the newbie. They can even be a little terrifying to the veteran who's trying to attempt a 30-color panel. And he's got a lot of experience doing simple stuff, but now he's got to do some really challenging things. So for me today, my talk is really bringing the conversation back around to fundamentals and the fundamental elements of a successful flow cytometry experiment. So with that, I'll ask for my first slide. So you can see here on my slide, there are a number of moving parts in a successful experiment, starting with experiment planning and design, panel design, experimental controls, considering how the instrument needs to be set up, how the data needs to be acquired and standardized, data analysis tools, the variety that are out there, and of course, always from start to finish, integrating technical and educational resources into the process. What I'm going to talk about fundamentally today is sample preparation, because none of those other moving parts mean anything unless we have a sample to bring to that instrumentation that means something that is properly prepared. So next slide, please. So what are the fundamental steps that on a daily basis I talk to my customers about as far as sample preparation? Starting with titering all of your reagents. There probably isn't a more important step to make sure that we're titering all the reagents to be able to get the best information out of our experiments. Blocking your FC receptors, using the proper temperature during your sample processing, filtering to avoid clumps, making sure we have those single cell suspensions, because flow cytometers require that kind of level of single cell suspension, including serum and protein in our sample buffers. And of course, everybody knows this, we need to use viability dyes. So next slide. Breaking these down into their individual parts, it can be pretty clear why these concepts are important to sample preparation. Titering our reagents allows us to determine the optimal staining conditions and maximize signal resolution, which is the ultimate goal of finding those populations of interest. Blocking FC receptors that are prevalent on a variety of immune cells is very important to avoid unwanted antibody binding. Using the viability dyes allows us to gate out dead cells that non-specifically bind antibodies and cause all kinds of consternation in your data. Obviously, if we're preparing live cell assays especially, but throughout the process of staining cells, temperature is a very important consideration to enhance the viability of your cells and maximize the staining conditions, especially when you're doing cell sorting, when you can be taking a longer time to actually process your samples. Adding serum and protein into your buffers, again, enhances the viability of your cells. And it also has FC blocking properties to allow you to get specific staining. And of course, near and dear to every core facility manager, single cell suspension is very important, vital to a successful experiment. We want to filter to avoid clumps getting into our cytometers, but even more importantly, avoid those aggregates from contaminating our DNA or our data analysis downstream. So there you can see these are fairly fundamental principles. Many of you may have already seen this. You may be rolling your eyes a little bit about, oh my gosh, how many more times do I have to hear about sample preparation? But I would imagine a number of our participants today are fairly new and young at this technology. And it's super important that we grasp onto those fundamentals in the sample preparation area, even before we get into the other fundamentals. So with that, I'd like to hand it over to our panel and our presenters to start the conversation about this. Thank you.
Thank you, everyone, for that excellent overview. As Jodie said, it's now time for the Q&A portion of today's session. And to kick this off, we do have a couple of questions that were submitted by registrants prior to today's event. So remember, if you have any questions for our experts live, please keep sending these in and we will get to them during the Q&A or follow up afterwards. So to start us off, we've got a question here for Jodie, and that is, how, when, and why should isotope controls be included in flow cytometry experiments?
So thank you for the question. This is probably one of the most perennial questions in flow. I can remember back in my first educational steps of doing experiments, that was a big topic, and we kind of just did what we were told because for a lack of better information and availability of controls, we just threw those isotype controls in there. So I'm going to also ask for some help from my fellow panelists in this, but I put together a couple of slides to help those who are not that familiar with the idea of what a isotype control is versus when they should be used or when they shouldn't be used. So isotype controls are used to identify nonspecific antibody binding. The important consideration you have to make when choosing an isotype control, if that's your decision, is you must match the heavy and light chain subclass to your specific. You also must match the fluorochrome protein ratio and manufacturing conditions to the test antibody. And these can be difficult to do, and it's kind of a circular argument because how do you match those conditions to something that's irrelevant, something that's not supposed to be stained on a cell? It's pretty hard to do. So the limitations, antibody conjugates can vary in the way they stain your cells in background capacity, the degree of aggregation of those types of antibodies, as well as other variables, making it very difficult to find a, quote, perfect isotype control. Single stain isotype controls do not take into effect spillover from other fluorescent channels because by nature of the negativity or irrelevance of the isotype antibody, they are negative for the fluorescence that they would carry with them. So they're not providing you a very beneficial background control. So when would you want to use an isotype control? Well, when you need to kind of assess the amount of FC receptor blocking, they can be helpful because if you're using an isotype control that's irrelevant and shouldn't be binding, yet the cells have a high amount of background, that can indicate something going wrong with unwanted binding of that antibody. You can use it to determine issues with non-specific binding, especially with intracellular staining, where the capacity for non-specific binding is actually elevated. And again, the isotype control antibody can indicate inherent stickiness of your cells pre or post-treatment, but they are not to be used as a means to subtract from specifically stained cells, whether percentage or intensity. Now, when you do not want to use them is when you're trying to determine the background of a signal of a sample. You don't want to use them as a gating control to determine positive or negative populations. If you have bimodal expression of your markers, say in the case of B cells, T cells, anything that clusters very obviously, you don't really need to have an isotype control. You have your own internal negative cells that are negative for those specific markers. It's actually better to use FMOs, or fluorescence -1 controls, as many of you may be familiar with, to account for the spillover background in channels as it relates to the other fluors that are contributing background to those channels. Now, some people who still want to use isotypes, I recommend to them, okay, incorporate them with your FMO. So where you're leaving one specific antibody out, substitute in the irrelevant isotype with the same fluorochrome attached. Of course, making sure to take care of those other considerations that we just talked about. You do not want to use an isotype control as a gating, I think we kind of repeated that, but as a gating control for determining positive and negative, or as a substitute for proper titration. I'm a big advocate for and stickler for properly titering our reagents. And always remember, even if you are getting background staining, if you do choose to use an isotype and you're still getting, you're taking care of all these FC blocking and you've done all the things that we've just suggested, you're still getting background. Remember, it could be from free fluorochrome binding. This is one of those conundrums that can occur that a lot of people are not familiar with, but there have been some cases where certain fluorochromes can bind certain types of cells, and you need to be careful about that. So I'd like to introduce in any other comments from my panel, fellow panelists, and see if they agree, disagree, or have anything to add to.
Yeah, I can reiterate a few of those points, I think, Jodie. I mean, you think you've covered a lot of what I would say to people as well. A lot of what we do in cytometry, particularly in the core facility, is all about making sure that people are empowered with the right knowledge. So, you know, you need to understand why you would want to use an isotype control in the first place. And, you know, we know that there are thousands of protocols out there, and people will grab them from lab members or off the internet without necessarily understanding why they're doing particular steps in that protocol. And that's important. You need to understand why you're doing everything in there and what impact that might have on your data. So I suppose the main reason why we'd use an isotype control is to try to control for non-specific binding of your specific antibody. And that is really dependent on the antigen binding site of the antibody. And, you know, an isotype is not able to provide that. There are, as Jodie was saying, there are some cases where an isotype is useful because not all binding is antigen binding site to the antigen. Some of it is FC receptor binding. So the use of an isotype might show you that your cells are sticky or expressing FC receptors. And there are things that we can do about that. We can put blocking steps in there to remove that non-specific binding. But they're not a really good control for what you really want to do. Now, the ideal control is something that you might not be able to do. It's having a knockout model. So you'd have cells that aren't expressing that particular antigen, but then you can stain with your specific antibody. And if you see any staining, that must be down to non-specific binding. And we certainly don't use the isotypes for gating, as you said there. I suppose there are other considerations as well. Sometimes people will use isotypes in intracellular assays because that helps you determine if there's any, not necessarily non-specific binding, but antibodies becoming trapped in there. So that again can help you with thinking about your experimental design or your protocol. It might mean that you need more washes or you need a different permeabilization process and so on. So I wouldn't say we'd never use an isotype control, but I think you'd have to think long and hard before you use that as something that would that really tell you what you want to know, which is the non-specific binding of your specific primary. And of course, controls are going to vary with every experiment as well. And sometimes you might only need compensation controls if you've got very good separation of your antigens, whereas other times you will need the FMOs, which are probably a better control anyway than the isotypes.
Yeah, this is.
Add anything, Bill?
This is Jody's slide is absolutely comprehensive. I would just say this is a confusing issue for a lot of people, because if you got into flow more than 5 years ago, you were told that you must use isotype controls, and they used to be strongly recommended to assess the issues that Jody and Derek just covered. And the manufacturers dutifully produced large, numbers of them for us to use. So the advice has changed a little bit. We still use them, but we use them in a more limited way. One issue too is the fact that many of us are now doing 12, 15, 18, 20 plus color experiments, and doing a large number of isotype controls is simply not practical anymore. But the list Jody has provided is absolutely comprehensive. And that's all I'll say.
So there's a second slide with a couple of references that I pulled from to make this slide. And of course, I solicited the help of my staff to garner those. So if you want to jot those down, those are great resources. We give them out to our new trainees as a habit. And there's also some additional information in there that Bill and Derek both alluded to, including instrument setup and other types of controls embedded in these references. So I highly recommend them. They're two of my favorite.
Thank you, Jody, for the slide, and thanks to everyone for that comprehensive answer. We do have a follow-up question from a live attendee who was wondering, why can't an isotype control be used for subtracting from specific positive percentages?
Who wants to take that?
Derek, kind of alluded to this earlier in your response.
Derek, would you like to?
Sorry, I lost my controls there for a second. Okay, I should be back now. Yes, with all our experiments, we want to make sure that we're always comparing like with like, aren't we? And generally with our specific staining, obviously we're looking at binding of an antibody to a specific antigen. But with our isotype, we're not doing that. We're looking at some sort of background. So we're not actually comparing the same things there. So I wouldn't normally subtract that. It's the same reason why I would not ratio a negative into a positive, because we're not comparing. It's the same thing.
Yeah, we don't really know what isotype antibodies bind to. We have this idea that they have no specific binding at all. And in fact, there are often things like plasmacytoma proteins that are monoclonal, but in fact, we really don't know what they bind to. And they may have a little bit of sort of incidental FAB binding to a cell. So they're not perfect by any means.
Bill, you bring up a really good point from a historical perspective on isotype controls from companies. Many of them originally came out in the really early 90s, even in the 80s. And they were replaced little by little as individuals used them. And sometimes they found that some of those isotype controls that we were using actually bound inside a cell. And there was some determinant that they saw. So then they were replaced over time. So you're absolutely correct. We don't know what the specific epitope is for any one of those. And again, if they're not matched up with the exact concentration to every single one of the specific antibodies, then the data sometimes is hard to interpret.
Excellent. Thank you, everyone. We'll move on to the next question, which is, so this is a more general question. So what are some of the key strategies for flow cytometry panel design? I don't know if, Bill, would you like to start?
I can start. I won't finish, but I'll start on this. Certainly. For us, one of the key strategies, and it doesn't necessarily involve using an algorithm or a table, but simply to make good decisions about designing your panel. You go through, you make a list of your antibodies. Some of your antibodies are phenotypic. CD4, CD3, CD8, they're either positive or negative. There's not much in the middle. Some of them are modulated. If you're looking at FOXP3 or IL-2 receptor, these are things that may be off, they may be on, and they may be on just a little bit. So you need to place those markers in relative spectral isolation from your phenotypic markers, so you don't get a lot of contribution from those very bright positives into your modulated marker. So make a list of your receptors from bright to dim, assign dim versus bright fluorochromes, assign fluorochromes that don't have much spectral overlap with one another. There's no easy way to do this. You just have to work through it, although hopefully we will have some tools soon that will be available. And then test your panel. And one of my favorite strategies is I get my little bottle of capture beads, and if I'm doing a 20-color panel, I label up a 20-color capture bead set of compensation controls and run it. Now, this doesn't tell you anything about how well the antibody is going to bind to a cell, but it's going to tell you very cheaply and very quickly if you have any significant spectral overlap issues between any of the fluorochromes that you're using. And then test that panel, then move to single color controls in real cells. We often recommend for high dimensional panels that people build up their panels, start with 4 to 6 colors, get that working nicely, then add in 8, 10, 12, 15, 18. It allows you to dissect out where your overlap problems are, which can be tough if you jump right into 20 colors. Trying to see where the spectral overlap is can be very, very difficult. So it's a lot of legwork. Ultimately, you can use the tools that are online for looking at spectral overlap. You can certainly consult the charts out there for fluorochrome brightness and a marker expression, but it's going to take some planning on your part.
Excellent, thank you, Bill. Derek, Jody, I don't know if you've got any key strategies to share.
Well, I think Bill covered the key strategies very well there, and this is what I would normally explain to new users. One thing that I do like people to think about when they're designing any experiment is to actually, even at the beginning, think about how you're going to analyze that experiment. Which markers are you going to use to define the cells you're interested in? Which are the key markers that you're looking for? Because sometimes that helps you with thinking about how you're going to assign fluorochromes to markers. You're always going to be asking a biological question, and the more you understand about how you're going to dissect that heterogeneous population, the better that's going to inform your experiment. So, as Will said, thinking about the antigen density, whether that's fixed or whether it changes when we do something to a cell or over time, and also thinking about whether you're looking at things on the surface or inside the cell, because to get antibodies inside the cell, you're going to have to fix and permeabilize in some way, and that fixation and permeabilization is going to affect both the cells and any fluorochromes that you've stained them with beforehand. So sample preparation is also very important. Jody didn't allude to that. That was the core of her talk. It's extremely important. It's one thing I didn't put on my slides, but people have to bring cells in an idealized format to the cytometer. And the more you understand about all of these things, the easier it will become. Sometimes it looks very opaque, especially if you want to jump in and do a 20-color panel straight away. Start slow, start on smaller numbers of colors, and then you get to understand both the biology, your protocols, and even the way you run the data and analyze the data at the end of it.
Wonderful, thank you. So we've got time for one more question.
Can I throw one more comment in there?
Oh, absolutely, Jody.
Sorry, Derek and Bill totally, they stole my thunder, but that's okay. I would just like to throw one more comment in, actually maybe two. The first strategy would be to start with attending a local or online fundamental training class or educational class. If you haven't been exposed to these kinds of things before and you're just starting into flow, there's no better tool than to look out there for some experts to buddy up with and learn the fundamentals that way. And then one thing that Bill almost said, I thought he was getting ready to, in addition to trying to avoid spillover as much as possible, I would also look granularly at the very important channels that you know are going to be kind of difficult to resolve because they're dim expressors or that you just don't have a lot of knowledge about them. try to avoid specifically spillover as much as possible into those channels, because that can be really a key strategy to allowing you to see those populations. That's all I wanted to say. Sorry.
Perfect. Thank you for sharing, Jodie. So yeah, we've got time for one more quick fire question. And this is spectral flow cytometry the future, and will it replace conventional flow cytometry? And then to add to that, how will this impact current studies utilizing conventional flow cytometry? So I don't know if Derek, do you have, would you like to start? Or perhaps Bill?
Sure. I think it ultimately is the future. It's already, if you look at microscopy as an analogy, spectral is now very common in microscopy. It evolved away from single bandwidths toward full spectrum collection. So everything is moving in that direction. Traditional flow is not at all dead. The conventional cytometers are not going away anytime soon, and we can do 30 plus fluorochromes on a conventional cytometer. But spectral cytometers have a lot more information in terms of compensation or spectral unmixing or deconvolution, whatever you call it. If you're looking at FTSE versus PE on a regular cytometer, it's a 1-by-1 matrix. If you look at FTSE PE on a typical spectral cytometer, it's a 64-by-64 matrix. So there's a lot more information in there, more granular, to use Jodie's term, that allow you to do better spectral separations. So as most of you know, we can certainly use fluorochromes that are closer to one another spectrally. Combinations that didn't used to be possible are now practical. But the same rules are going to apply. There's nothing magical about it. You still need to design a good panel. You still need to avoid certain fluorochrome combinations and certain receptor labeling combinations. That's not going to change at all. It may be a little more forgiving, but we're still going to have to follow the same rules in terms of panel design. And as Jodie said, we still have to have good samples. If your samples aren't any good, your data is not going to be any good, no matter what technique you run it with.
Fabulous, thanks, Bill. Oh,
yep.
Yeah, I was just about to ask Jodie.
I'm new as far as a user to spectral flow. I've been doing a lot of reading in the background, paying attention to what's been going on in the community, going to a lot of seminars and talks for people who've got a lot more experience in this area than I have. So I've recently just acquired my first spectral, and I've had the benefit of taking a look at my Symphony data. The same samples run on the same day from the Symphony to the spectral. It's been quite enlightening. I can say with limited experience that I am not seeing much difference so far, but I've only had a couple of projects that I've been working on, so I can't, I'm not going to tell a full story out of 2 projects. I will say that I'm hearing disturbing comments from the average user who comes along in my lab and says, "Oh, we have a spectral analyzer. I don't have to do compensation anymore." It may not be called compensation by the manufacturer that's selling that instrument, but it's still a form of unmixing or deconvolution of the signals. Yes, you still have to do your controls. You still have to prepare your samples properly, and probably even more so. What I'm understanding is any bad habits you've been getting away with on conventional flow cytometers can actually be exacerbated on the spectral analyzer. So be prepared for the rigors that you're still going to be expected to follow. 5 years from now, we may be having the same kind of talk, just a little bit more focused on spectral cytometry, where people are going to be going, "Well, why can't I do this? And why should I have to do that?" It's still going to be flow cytometry in the end, is my opinion. I guess that still remains to be revealed to me, but that's the way I see it so far.
If I can just add a little bit to that, that's absolutely right. And with regards to the second question, your compensation or unmixing or deconvolution controls, I think, are even more important in spectral, because you're looking, on a high-end spectral cytometer, you're looking at all of the for every fluorochrome. Doesn't matter what laser excites it the best, you're looking at all the spectra for all the lasers. So all of that ultraviolet autofluorescence and violet autofluorescence that we can sometimes not pay attention to as much in traditional flow is very there in spectral. So if anything, I think your controls you have to be even more rigorous about controls. Because Jody's absolutely right. It's still compensation. It's just compensation over a broad spectral area. We give it a new name, but the mathematics behind compensation and unmixing or deconvolution are the same. It's just a more fine-grained compensation. So the same rules apply, and we have to take the same factors into account when doing our controls and preparing our samples.
I'm like a kid in a candy store. I've been amazed at the learning curve I've gone through with fluorochromes. I mean, the fluorochromes I thought I knew all these decades that I've been doing flow have been newly revealed to me on a spectral analyzer. So that's something to be considerate of as well. You're on a traditional cytometer, you're looking at a bandwidth of signal, and you're subtracting that signal from the others that it's spilling into, but you're really still accounting for that signal in its own channel. Well, now you take FITSI and you can see all these different signals across the entire spectrum. It's very eye-opening.
Wonderful,
Absolutely.
Thank you. Derek, do you have...
I was just going to add a couple of things. Yes, it's a more philosophical question, I suppose. But is it a future, and is it going to replace conventional flow cytometry? Well, you could argue that all flow cytometry is spectral. It's just not full spectral, as we're looking at now. But I suppose this is what we're looking at in the field, isn't it? It's not necessarily the future, but it's maybe the next big thing that will help us maybe clean up things a little bit. We're always looking to different fields, aren't we, to help us in pushing flow cytometry forward, whether that's chemistry in terms of making new fluorochromes, or whether it's astronomy when we're talking about looking at low levels of light, or whether it's bioinformatics when we're looking at multi-parameter data analysis. We're always trying to push the boundaries a little bit further all the time. And spectral, or full spectral flow cytometry is something that's certainly going to be useful over the next few years, I think. But as both Jody and Bill have said, it's not a universal panacea. We do still have to think about panel design. There are limitations, and we need to be aware of those. It's all about knowledge and empowering the end user.
Wonderful, thank you. Unfortunately, those are all the questions we have time for today. So I'd like to hand back to Bob.
Well, first of all, I'd like to say thank you to everyone. It's clear that moving into an area of spectral is bringing lots of questions and lots of perspectives by individuals, and I think it's one of the key takeaways that we have. It reminds me a little bit of those days around 2000 when there was a large change going from an analog instrument into a digital instrument, and all the changes and the perspectives that we had to learn. So as Derek said, and as Jody said, we're going to have to learn as a community. Some laboratories are already transitioning into that technology and are getting great results from what they see. But at the same time, they have to learn to set up their controls. They have to learn about what colors can be used together, what colors can't be used together. And I think that's just part of this overall process that we're going through in terms of fundamentals. I'd like to go back, into some of those other fundamentals too, because they always come back. And I think those elements of how we work with that sample, as Jody, set up for us and detailed out the controls that we have to put into a flow cytometry experiment. The challenges, as Bill said, that we have as we move into higher parameter flow, and we have fewer and fewer cells to work with, but we're trying to get out a tremendous amount of data. And one of the takeaways that was clear today was that we didn't step into that area of data, and how do we analyze it, and then how we control to make sure that the underlying flow experiment that we do when we move into all of these new TISNEs and UMAPs and all the different ways that we can analyze data, underneath that it's really important to make sure that that the structure of the experiment was done in an appropriate way so that we can measure it, effectively. So I think all of those things are really, really important to us. I'd like to also take the moment, though, just to direct everybody's attention to some of those areas that we have. Some of the tools that are on the website that are there for us and how we design panels. Some of the other YouTube snippets that we have out there that we've had fun with, and we collect all this information from so many different directions and perspectives that all of you share, that we try to do everything we can to bring fun into what we do, to bring the learnings into the thousands of people that use flow cytometry. And one more very, very important thing is that deeper and deeper data is what we search for all the time, a greater understanding of really the fundamental of the exact experiment we have. And today we're bridging those technologies. It's not only flow, it's moving into single cell work. All of these things need great controls and great understanding. So I'd like to direct everybody's attention to a new webinar. Dr. Mirko Corselli in our program has really something new for everyone. And I think everybody, if they can tune in on May the 13th, are going to see something very, very different as in respect to the spillover spread matrix that we've all been using for a long, long time. I'll leave it with this. There's certainly something in there that needed to be corrected, and Mirko found it. So I hope everybody tunes in for that webinar, and we'll have fun with it too. For everybody at BD, for our host here today, I'd like to say thank you to everyone, and especially for Jody and also for Bill and for Derek. Thank you for your time, and thank you for your perspectives. And we hope to see you very, very soon. Thank you very much.
Yes, so a huge thank you to you as well, Bob, and of course our panel of flow cytometry experts for today's informative discussion. And thank you to everyone joining us online today. I can see that there are a lot of questions for our experts still coming in, which is great. So we will follow up with you after the webinar if we haven't managed to get to you today. Also, please feel free to email me at editor@selectscience.net with any further questions. If you'd like to share your attendance or provide feedback using social media, please feel free to mention us using @SelectScience or #SelectScienceWebinars. Remember, you can also download your certificate of attendance and access the resources Bob kindly highlighted in our related resources tab. And if you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days' time. So goodbye, everyone, and thanks once again for joining us.